We are studying the structure-function relationship of glycolipozyme named MPIase, which is involved in protein localization. Proteins are usually synthesized on ribosomes in cytosol. On the other hand, eukaryotic cells contains organelles compartmentalized by biomembranes. Therefore, proteins localized in respective organelles must be sorted and transported to the correct destinations. A simple organism, E. coli, is widely used as a model organism, because the fundamental mechanisms of protein localization are thought to be conserved among all the living things. Let's see the surface structure. E. coli possesses two membranes, outer and inner membranes. The space between two membranes is called periplasm. Some of proteins are localized in these three areas. They are transported after the synthesis in cytosol. This figure shows how proteins are integrated into inner membrane and translocated across the membranes. You can see that many proteinaceous factors function in a concerted manner. We found that a glycolipid named MPIase is involved in all the processes shown here, by biochemical analysis. We determined the structure of MPIase. The sugar chain composed of repeated unit of 3 N-acetylated amino sugars is connected to diacylglycerol through the pyrophosphate linker. We named this glycolipid MPIase because MPIase catalyzes protein integration into membranes. MPIase receives newly synthesized membrane proteins on membrane. After MPIase modulates the surface structure of membrane, substrate proteins are integrated into membrane. Then, MPIase catalyzes the next cycle of integration. YID-C is known to be involved in membrane integration. We found that MPIase cooperatively works with YID-C. At first, MPIase drives membrane integration, and then the substrate proteins are transferred to YID-C to complete integration. Previously, we demonstrated that Sec-G, a subunit of Sec-YEG, undergoes the topology inversion cycle to stimulate SECA-dependent preprotein translocation. Surprisingly, we found that MPIase is involved in Sec-G inversion. The reason is that MPIase transforms the dimer structure of Sec-YEG into an active form like this. Only in the side-by-side structure, Sec-G inversion occurs, achieving the maximal activity of preprotein translocation. We also found that the expression level of MPIase is significantly upregulated in the cold. So far, it has been known that protein integration and translocation are intrinsically cold-sensitive. Therefore, MPIase suppressed such a cold sensitivity. In summary, MPIase, we discovered in the biochemical studies, is a multifunctional glycolipid. MPIase can catalyze membrane integration of simple membrane proteins by itself. Moreover, MPIase functions cooperatively with YID-C, Sec-YEG and TAT-ABC. We also found that MPIase is upregulated in the cold to suppress the cold sensitivity of protein integration and translocation. Recently, we found that MPIase homologs are widely conserved in mitochondria and chloroplasts in eukaryotic cells. The glycolipid MPIase with unique functions could be applied in several ways. We have succeeded in reconstitution of functional integration of membrane proteins. This reconstitution system can be applied to the high-throughput analysis of membrane proteins. The proteoliposomes with the synthesized membrane proteins are ready for functional analysis, which enables the drug screening. Next, the development of cold-resistant plants would be possible by modulating the plant homologs of MPIase, because MPIase suppresses the cold sensitivity of protein export. Finally, elucidation of the mechanisms of aging would be possible by analyzing mitochondrial MPIase in animals, since mitochondria possesses MPIase homologs that are involved in protein sorting and integration. Thanks very much for your attention.